Okay, everyone, good afternoon and welcome to main stage 4 of the International Rare Disease Showcase. Really great to be here, and I hope you've all had a really enriching and interesting morning of sessions, and are looking forward to even more enriching sessions this afternoon. So before we kick off the session, you'll have seen this slide many times before, but it's really important that we thank all of our sponsors for the showcase today. So that's our corporate partner Helix, our gold sponsors Pulse InfoFrame, Bionicle EMAS and Apparito, our silver sponsor Eusa Pharma, and our bronze sponsors Gilead, PTC Therapeutics, Roche and Rhythm Pharmaceuticals. So today's session is called Harnessing the Power of Technology to Drive Diagnoses and Understanding. And obviously we know how important diagnoses are in the rare disease space. We can't understand the true scale of rare disease either in our own countries or internationally until we know how many people have the rare disease in question. So it's so important that we're able to streamline this process to truly understand and gather data on the amount of patients that we need to be helping worldwide. So hosting today's session, you've got me, I'm Philippa, or Phil, I'm the project's coordinator at Beacon, and I'm joined by Rick, our CEO, who you will have seen a bit over the past few days as well. So in this session, we're going to be exploring some really interesting approaches to data and diagnosis, and we're going to be looking at how patient advocacy and collaboration can drive patient empowerment and ownership when it comes to health and data, the utilisation of technology to uncover new approaches to data and diagnosis and improve upon old ones, and learning about the crucial role that patient groups can play in this process. We've got some really interesting talks not only on how we can empower patient groups and patients through data and different platforms, but also through approaches to things such as newborn screening and the developments that's happening in that space as well. And I just wanted to bring up something really interesting that I thought Kelly said earlier, and she said, why are we not giving medical information to individuals in a way that empowers them? And I thought this session might be a really good way to answer that question and to provide a little bit more inspiration around that statement that Kelly brought up earlier. So just to introduce some of our expert speakers today, we're joined by Marie from SMA Europe, Mikael from PerkinElmer, Edith from EURORDIS, David from the European Sickle Cell Federation, and Pablo from Foundation 29. And I'm really excited to hear what they have to say on this topic today. So without further ado, I'm going to pass over to Marie, who's going to be talking about a European Alliance for Newborn Screening in Spinal Muscular Atrophy, and really looking at that campaigning and advocacy approach to making change in this area. So over to you, Marie. I'm really excited to hear what you've got to say.
Hello, everybody. Just to share my screen. So I'm just wanting to present you a very amazing story about the Newborn Screening Alliance. My name is Marie-Christine Ouillade. I am a mother of a lady who is 27 years old, who is living with SMA type 2, and also a volunteer at IFM Téléthon, who is a very large organization of patients with neuromuscular disease, and board member of an alliance of SMA Europe, covering 23 now countries in Europe. And last year, they decided to nominate me as the Newborn Screening Alliance Chair of the Steering Committee, and it was a very interesting job and interesting journey. Why did we need, I think I have lost one, no, but we do need a newborn screening for SMA. It's very important. It's a rare disease, but each year in Europe, we have 700 of children born with SMA. And the newborn screening in SMA is quite easy to diagnose. This is a monogenic disease, so very easily we can have a diagnosis and we can have an early diagnosis. In parallel, newborn screening is a very, very devastating disease, especially for the severe form, and time kills motor neurons. So we have to diagnose the baby as soon as possible to stop the death of motor neuron. And you can see on my slide that it's a very, very devastating disease, especially for type 1, but also for type 2, who lose the capacity to work before the age of 2 years old. So it's also a lifelong with and unable to walk, was also in a wheelchair, on an electric wheelchair. What we can see and what we learn from the past is that if you treat the baby at birth, especially for the mild form, they have a normal development, at least till the age of 5 years old, because the oldest now treated at birth are 5 years old and are very well, and they work, they have a normal life. To compare to the baby treated after the first symptoms, that means that part of the neuromuscular and the motor neuron are dead, and there are still difficulty to work or sometimes unable to work at the age of 5. So it's a very, very important to treat as soon as possible and to diagnose, of course, as soon as possible. But if you diagnose this with symptoms, it's too late for this baby. So we decided to create this alliance after an important meeting. SMA Europe has a very important scientific meeting in Évry 2 years ago now, with more than 600 clinicians or researchers on the field, and all the presentations go in the same way, saying that we have to diagnose as soon as possible, and it's urgent to treat baby, and it's urgent to have newborn screening in Europe. We are a patient organization, and we say we must go forward and we must gather and put together all the stakeholders of the field to do this. And what we discovered when we ask for newborn screening is that in most of the country, you have to apply country by country, and in some country, region by region. And newborn screening, the health authority always asks for the same thing. So you have to duplicate the dossier in each country, sometimes 10 times in the same country, for example, in Spain. And patient and also all the stakeholders of the field have to do the job many times and the same jobs and gather the same data. So we create the alliance and we go to many stakeholders. Of course, we need to have a professional support. We find a professional support. And then we try to put together on the same table on the most important partner. There is, of course, patient organization, SMA Europe covering part of Europe, but also EAMDA and EURORDIS will be part of the alliance. We also put on the table some very important network. The most important one is EURORDIS, who is the ERN for neuromuscular disease. And you see also Treat NMD, Screen for Rare, and so on. We put together also some university who are far advanced in SMA and in SMA screening. We put the pharma, the different pharma company who have a treatment in SMA. What we have to notice is that whatever the type of treatment we have, we need to treat as soon as possible before the death of motor neuron. There's no difference if you get one or the other pharma, we need to treat very early. And then we also put around the table the screening kit manufacturer that can support us on a technical point of view. It was quite easy, I will say, to have all these people around the table, and we were able to make the difference and to make the start. What was also the decision of SMA Europe is to have a steering committee. And in the steering committee, we have to have patient organization and academic. The organization is driven by a non-profit organization to be sure that they are compliant, and it will be very focused on the newborn screening for SMA. Our vision is quite easy to understand. We want to all the baby to be screened over Europe, fully reimbursed by public healthcare. It's very, very important to have this. And our objective is to have all the babies in Europe screened for before 2025. So it's quite soon now. We have a good start. At this time, we estimate that in 2021, more or less 15% of the baby has been screened. Probably at the end of this year, it will be around 25%. So there are still some work to do, but we start. What we do? First, we discover that we need to go to the National Health Authority, and for that, we produce some a flyer and some poster to be able to go to the different National Health Authority, and also we translate it in different languages. For example, the flyer is available in around 10 different languages now. And we produce a website where you will find all the relevant information as soon as we were able to produce this information. So it's the website was the place to find all the documents. If you want to go to the website, you have the address of the website. But it's not enough. And what we produced was very, very effective for the patient and all the people who request newborn screening. Is a white paper collecting all the information, or most of the information requested by the national agency. What is SMA? Why SMA meet the Wilson and Younger criteria, a small health economic study, all the ethical agreement, mostly issued from EURORDIS paper. And this paper was very, very useful for many countries to apply for newborn screening or to start the discussion with the national authorities. Of course, the most important thing at this time, because most of the agencies are focusing on this criteria, is to show that we are able to meet the criteria from the Wilson and Younger. And as you know, SMA will be a very important health problem. Baby die with SMA. There are accepted treatment. We see the 3 different accepted treatment. It's quite easy to diagnose and is a genetic test on only one protein. And so it's very easy to diagnose with this. It's the SMN1 gene. There are very early stage of the disease. Half of the baby show symptoms before the age of 6 months. There are, of course, this test, and you will see with Mikael that we can produce a test for SMA. It's acceptable for the population. It's mainly requested by a lot of part of the population. There's a very good natural history. Agree policy about whom to treat, even if it still be some very interesting discussion. And the cost of case funding is quite balanced regarding the disease and the difficulty for the healthcare system to follow this baby. So it's a very easy to advocate, but not so simple to have an agreement. If you look at the map today on our website, you will see the map with the status. You will see that there is only a few parts of the Europe who have newborn screening program in place. At this time, is mainly Germany and Sweden, and also we have some in Sweden, and we have now Belgium, who is in green from the 1st of this year. And a lot of places have active pilot. And that's also a problem for us, because each country wishes to have its own pilot. Even if the result for the pilot are the same in each country, They do not agree that the pilot we do in Germany or in Belgium is valid. They want to do their own pilot at home. What we are going to do in the future, we have produced a webinar who's now available on the website because we have a lot of questions about the different technique of screening. And it was a very interesting webinar, and you can see the webinar. It is available on our website. We are now working on a very large cost efficiency studies, focusing in different type of country. It's very not easy, but we have some cost efficiency study in western part of Europe, for example, Germany or Belgium. But we need to have data on eastern part of Europe. Poland is producing one, and we are going to continue this. What also we discover that is quite challenging is to clearly understand the pathway to request newborn screening in a country. It's absolutely different from one country to another one. And this is also a very challenging part for advocacy. And then we do, as I do today, a communication in the different congress to make awareness about newborn screening, especially newborn screening on genetic disease. We go also to some European deputies, some MIP, because we thought it could be relevant to have a newborn screening information at EU level and not at country or regional level. What I want to finalize to my talk is our experience able to be duplicated for other disease? Of course it is, but I think it's not the best way to treat, except as we are in an urgency to treat immediately this baby. I think the best way is not to go via such alliance, disease by disease. Something we learn is that the Wilson and Younger criteria, maybe they are a little old and they have to be reviewed. In many cases, there's no cure for baby with a severe disease, but a good standard of care at the earliest stage of a disease can change the course of a disease. So even if you do not have a treatment, it could be very interesting to have this. And also to make the evidence that the treatment on newborn screening is efficient, you need to have baby screen to include in the different trial. And so you need to have some newborn screening earlier than a treatment. What we see also, it's a very, very important use of time and effort to go country by country, region by region. What we suggest to our deputy that we have in face is that it will be relevant if Europe or maybe EMA can give a scientific advice about the rationale for implementing newborn screening in a disease. It will be very, very helpful to have a sort of recommendation from EMA saying this disease is important for newborn screening. And so when you go to the national authority, you just have to make the proof that is probably important to be reimbursed in the country, more or less as a drug at this time. And what also we think is that more in the future year, there will be a lot of genetic disease available for treatment. And it's very important at this time not to only screen disease by disease, but to screen many diseases in the same screening shot. It's very interesting and important. I know that some industries start to create multi-disease testing, but it's very, very important to to have this. And we have also the project of Screen for Care and PDME project who can go there. And just, I want to share you one thing we are very proud this year. We will be nominated and valid for awarded for the company patient engagement, and it's a very, very important for us to be on the top of this. We will be able to include in the same table, in the same alliance, industry, academic, clinician, and patient, and work together with the same objective. It will be, for me, I was very proud to perform, to do this, and very proud to receive this award from EURORDIS. And here you can see all the members of the Alliance at this time, just to show you how important it is. And to be clear, the pharma industry, the screening industry, not only provide us some money, but they provide us data. They support us in writing, for example, the white paper, in providing educational program to the SMA Europe member. So it's very important to be together to fight regarding this disease. So if you have any question, or maybe we can go ahead. Thanks.
Thank you so much, Marie-Christine. I really thought that point you made about even if there's no cure, helping these patients and families and carers from the very first point is really important. I think we're going to hear a bit more about that going forward. And also we're going to hear a bit more about the Screen for Care programme and how we can streamline these screening processes more in this session. So thank you for setting us up with that really brilliant talk. And we'll have some questions for you at the end, I'm sure. So I'd love to now introduce Mikael to the stage, who is the neonatal business lead at PerkinElmer, and he's going to be talking about expanding newborn screening and more testing needs easier workflows. So thank you, Mikael, and over to you.
Thank you. All right, thank you so much for the opportunity to come and discuss this topic today. My name is Mikael Hjort. I'm the neonatal business lead. I'm representing reproductive health from PerkinElmer. I would like to thank Marie-Christine to begin with for the job that you have done with the SMA Newborn Screening Alliance, and I think you covered many of the topics that I also was going to have in my own presentation. So surely really helpful for me. I wanted to take the angle and really ask that, is it so that technology allows us to screen for more, or is it something additional that is still required to get more disorders in the screening panels? To start with, saying that really the objective of the population-based newborn screening is having early detection of asymptomatic infants. That is where, if there is effective treatment available and early diagnosis is critical. So screening started with phenylketonuria in the 60s, and then in the 90s, we introduced tandem mass spectrometry as a technology, which allowed then to screen a lot of more disorders. So this had a lot of benefits in diagnosis and treatment, but at the same time, it led into unintended consequences that are overdiagnosis, unnecessary treatment, and some parental anxiety. And what is happening now is that we're moving from these immuno and enzymatic assays and tandem masking to molecular and genetic assays. We're moving into NGS as a part of second-tier testing, but even as a first-tier testing. So it is certainly a something like a moving target, and technology allows more things for us to do. PerkinElmer as a company, so we in reproductive health, we focus on both on maternal, fetal health side and so prenatal screening. But today I'm going to talk about the newborn screening side. So we are the global leader in newborn screening, and we we have aided in screening over 600 million babies worldwide. Annually, we screen 50 million babies in over 100 labs. So we can count that every day 70 babies get a better chance surviving their disease state by using these technologies. But I want to start today following by Marie-Christine's talk on SMA and talk about more about SMA and how I see SMA in newborn screening. So thinking about spinal muscular atrophy, the question to my mind came that does technology really drive the newborn screening of SMA? Because if we think about the evidence, so we have papers saying that, okay, we can actually screen for SMA. There's a health economic analysis saying that it makes sense. We have technologies such as quantitative PCR. We have modified LNA, modified nucleotides, which helps for the specificity of binding to SMN1 exon 7. So that's really a high detailed technological fact. We have chemistry development, and we have a factory. So we have different technologies that is required, but is that really driving it? Or is it the SMA treatment and the advances that, like we heard that, yes, there are 3 approved drugs. So is that what really drives the fast implementation of newborn screening that we are now seeing, even though we go from country by country, expanding the SMA in the newborn screening panel, it is moving fast on a global perspective. So for those who are not that familiar with the newborn screening workflow, it's important to sort of emphasize how it actually happens. So we have a sample collection phase at the hospital. Typically, we collect the blood sample from the healed prick into the into the filter paper. We have a demographic data entry part, and then we go into actual analysis where you have a piece of the sample taken, punching out from the card. You have an analysis with different kind of methodologies, possible retesting of the sample, and then reporting. And then you continue in the confirmation, perhaps genetic counseling and treatment. So the question again is like, when we talk about newborn screening using different technologies, is it important? And is it actually this part in the middle? Or is newborn screening a wider perspective? Is it a service? And the answer is that it's all of this, and it's more basically when we look at technology perspective, we're just in this box looking at what kind of assays can we actually do. But that's not enough to actually be able to implement and to gather the evidence to bring in more disorders into screening panels. So really, you need to think about broader perspective, and you need to be able to understand what concretely happens in the newborn screening environment. If purely looking at the solution that we had in place for SMA screening, so we have a platform that is called, it's called EORDIS, and we decided to do an automation system for it. So it has automated DNA extraction of the DBS. You can do multiplex detection of spinal muscular atrophy, severe combined immunodeficiency, and X-linked agammaglobulinemia. It's a total CIVD solution. And Marie-Christine was saying that it's easy to screen and easy to diagnose. In a way, that's true. And on the other hand, it's not, because for the newborn screening labs, this would be the first or second molecular or genetic assay to implement. And there might not be skill set to do this, or there might not be the laboratory facilities required to do this in their lab, in their spaces. So what we thought from here is, excuse me, we need to take it further. We need to look at how we can reach more patients. And how we solve this issue now and to make it further is that actually we are not bringing more technology. We're not doing any more tricks. We're simplifying it. So we're taking down the level of actual automation. There is minimal intervention by any technicians. So you have minimal amount of sampling steps. You have minimal instrumentation. And that's in a way the key, that if we want to expand this kind of testing into a lot of different countries and regions and laboratories, this is the way that you can actually reach more babies for SMA screening. And we end up in a solution that looks like this. And the key features is that you have a really simple workflow with a less than 2 hours turnaround time. It needs to be full CE-IVD, something that is easy to train and, and ship and store, and doesn't really require even in like a specific molecular lab. So this is like an example of yeah, we use a certain technology. We have tools to do molecular newborn screening assays, but still, if we want to take it further, we need to get the customers' experience, and we need to develop in the more simplified system, which allows then a further expansion into different regions and countries. I want to give a couple of more examples because this is only SMA. This is only PCR testing. We cover the whole range of newborn screening in terms of analytes, whether that's your immuno and enzymatic testing that can be done with a manual platform or a fully automated GSP platform that is widely used globally. We have mass spectrometry, we have different analyzers and reagents for that, and we have also solutions for, as said, for qPCR or even for NGS. And we have the software solution to go from, so in a way, we can reach from sample to result. And SMA was the example of using qPCR. And yeah, we have also, I want to mention that we have worked together with different stakeholders and worked together while developing the assays, because we obviously rely also on getting the positive samples to be able to do the validation for the assays and for the methods. Now for the mass spec and newborn screening with tandem mass, so the key is that you can actually screen for many diseases using one spot and one assay. And it currently focuses on disorders of amino acid and acyl carotene metabolism, and also LSD, so lysosomal storage disorders. And key is that this is really cost efficient when you can actually multiplex and you get a huge amount of data from one single sample. What we need to remember that we are screening for better outcomes. We're not screening to get more data and to get more confusion. So really the things that is in the screening programs, like was already mentioned, like there needs to be a suitable screening assay, there needs to be a treatment, and it needs to be effective, it needs to be something that you should treat pre-symptomatically. So not the mass spec allows us to do a lot of stuff, that it doesn't mean that we should be doing a lot of testing just purely for the technology's sake. But for example, in the Pompe disease, as an example, you are able to do using a 6-plex lysosomal storage disorder assay, which one of them is Pompe. And the key here is that you can actually identify these affected as early as possible. You get the high survival up to 100%, and allows to do provide the reproductive choices for any preventive actions going forward. Another exciting example of how actually treatment and developing screening assays are going hand in hand is metachromatic leukodystrophy, MLD, which it is a fatal genetic central nervous system disorder. And here, you can actually screen for MLD using mass spectrometry. And there is a therapy available and approved by EC. And again, in this case, the need for the screening assay comes from therapy, and the information of what is the correct timing to actually develop the screening assay and to have it accessible to find those patients, get those into treatments, has required, of course, collaboration between different stakeholders. My last example here is what I want to highlight is Duchenne muscular dystrophy, DMD. And what I'm showing here is a picture of our main workhorse GSP automated newborn screening analyzer, which has a really wide coverage in newborn screening laboratories all around the globe. And so Duchenne is the most common and severest muscular dystrophy, which causes progressive muscle degeneration and premature death. And there is a assay available from the heel prick from DBS format, where you can detect CKMM from the sample, and by that way, you can screen for Duchenne muscular dystrophy. And the reason why I chose this example today is that we have the assay, and it has been approved by FDA. And in New York, US, there is a patient advocacy group called BPMD, and they are purely focusing on DMD. And they chose to partner with us since we had the most advanced creatine kinase MM test available, and it was already FDA approved. So they founded out this unique consortium, which comprises of 5 different pharma partners, us as a screening partner, they as the advocacy group, connections to State of New York Department of Health, connections to birthing hospitals, and then also to ACMG and NIH. And we have been running a pilot for 2 years for 37,000 newborns. Also, we've done confirmatory, they have done confirmatory NGS testing with our products. And really, what this pilot has resulted in is to prove the net benefit of DMD newborn screening. And in the end, now these results will be used for RASP nomination that will go in for March this year. And hopefully this will end up into full evidence review, and in the end, inclusion of DMD into the recommended uniform screening panel. But this is a good example of how different stakeholders are working together at the same time to actually gather the evidence data that is required to go for the evidence review, to get the acceptance to add the screening, this new disorder into the panel. Lastly, I really want to emphasize in a clear way that the assay is only a small part of the newborn screening service. It's a crucial part, but it's only a one piece of it. And if we think about bringing out new assays, bringing out new technologies, that's important. But as important is also for example, the speed and accuracy and the ease of use of those new technologies. We can harness the technology to have better outcomes for patients. And really by collaborating with all of these stakeholders that we have mentioned, so that allows to get out with right product at the right time. So the call for action is for everyone to reach out to us, discuss more about newborn screening, discuss about your needs and any possible collaboration. Thank you.
Thank you so much, Mikael. I really enjoyed how you emphasised that whole workflow as being kind of part of a care package, not just kind of the testing part of it. Every part of it can make the patient and family experience better. And I'm sure we'll have some questions for you a bit later on as well, but thank you so much. And we'll see you a bit later on in our question session. And I would love to pass over now to Edith Gross, who's joining us from EURORDIS. She's the Health and Social Science Senior Project Manager, and she's going to be talking to you a little bit more about Screen for Care, which Marie-Christine introduced earlier, and utilising genomics and advanced digital technologies to shorten the path to the diagnosis of rare diseases across Europe. So welcome, Edith, and over to you.
Well, thank you very much, and thank you to Beacon, and congratulations for your event, and it's wonderful to be here. I'm going to present today Screen for Care, but also I'm mostly from the part of EURORDIS part in it, which is, I'm happy to say, quite substantial. So let me share my screen with you. Okay, so Screen for Care is a new project that began in October 2021. I joined EURORDIS around that time, and I lead this project from EURORDIS perspective along with several of my colleagues. And we have a little time today, so I'm going to be a little bit quick and not very sensitive to a lot of details, but I really want to give you kind of an idea of what's going on in this project. So what is Screen for Care? Screen for Care is an IMI project, meaning we have partners from industry, from medium and small enterprises, and we also have academics. So as I said, we started early October. We're still in the starting point in many of our tests. We're up for 5 years, so we expect to be finished around 2026. We have a budget of 25 million euros. We're active in 14 countries with 35 partners. So it's really a big project, and I'm hoping to give you kind of a taste of it today. So these are the partners. I'm not going to go through all of them, but just to give you an idea. So we have industry partners such as Pfizer, who is leading the side of FPA on the call, and we have academics as well, and we are here as EURORDIS representing the patient voice. The project is very complex. It is a 5 years project. As I said, we have a lot of work packages which are separated into tests. I'm not going to talk about all of this. I am going to talk about small aspects of it, and especially where patient voice is most present. Okay, so instead of going through this amazing grid, I'm just going to summarize Screen for Care for you in a very easy way. Really, the idea is to provide better and earlier diagnosis for rare diseases, and we're using innovative approaches based on two main pillars. One of them is the genetic newborn screening, which kind of relates to what we've been discussing earlier, and the other one is the artificial intelligence and use of machine learning through health data, which kind of relates to the discussion I think we'll be having later on. Okay, so I'm going to talk to you about those two pillars with a certain focus on the first one. Okay, so what are the goals that we've identified for the newborn screening? First, excuse me, okay, we want to explore early diagnosis. We want to screen asymptomatic newborns. So really, we're talking about newborn screening. We're going to use next generation sequencing. We're talking about panels that we're going to be using. The idea is eventually to expand the spectrum of rare diseases that should be included in screening. And here I really connect with what Marie-Christine mentioned. There is here an idea to go further and not go disease by disease. Complementary to that, we will be using whole genome sequencing to identify diseases where we have early symptomatic infants who are not diagnosed. Okay, how are we going to do that? So we have a plan to screen 18,000 newborns in 3 countries. So we have Germany, Italy, and the Czech Republic. And we're going to have 6 centers where the screening will be done. Then the sequencing will be done separately, and the results will be returned to the patients. In the newborn screening, we're using panels, and we've identified 2 kind of sets of diseases that we're interested in. And I think here we have an innovative approach. So we have the core sets of diseases, which are still going to be 80, which is significantly more than is screened anywhere in Europe. So 80 diseases will be on the treat panel, which are diseases that follow more closely the younger criteria, the Wilson-Younger criteria. So we're talking about diseases where we know there is a treatment that is available and so forth. And here we have the actionable diseases. So actionable diseases are diseases where we believe that even if there's no cure, as Marie-Christine mentioned as well, we do believe that knowing and some kind of intervention, even if it's social support, is useful and beneficial for the patient and their family. EURORDIS is much involved in the actionable panel. We're in fact leading the work on that. And it is a very complex work. So how do you define actionable disease? We have taken the term actionability from work that we've done before in EURORDIS with our partners. And we're really asking ourselves, so what are these actionable diseases? How do we define them? How do we select them? There are immense ethical complexities to take into account. As we know, in terms of NBS advocacy, so newborn screening advocacy, different bodies have different perspectives. If you read papers on the subject, you'll see authors have very different positions. Depending on the definition, all diseases can be actionable. So if we say that knowing is good, then here all diseases are actionable, we should test them all, or none are actionable. So if you only define the action as a cure, you'll find yourself with a very small set of diseases. There is a continuum between the 2 panels, so the treat panel and the act panel. So we've made kind of a separation between the 2, with 80 on one side and about 200 on the others. But really, the conceptual link between the 2 is very complex. So, the considerations are highly dependent on the national and regional context. So different treatments are available and different types of care are available very differently in different countries. So it is very important to be aware of what is available for the patients, because again, this is a patient-centered initiative, and we believe that if we want to find out about a disease, we also need to be aware of what is available for the family. Another issue with that makes it very complex is the fact that science and technology are ever changing. So having a list of disease that is set on what's happening in early 2022 will mean very little in a few months, and definitely in a few years. Still, we're taking this approach, we're taking this challenge, and we've developed an approach. The idea is to discuss ethics and science and build a sustainable and adaptable model that will be based on well-developed criteria and a strong empirical basis. So here we have the idea that it should be sustainable, it should be adaptable, because we're talking about a pan-European approach, and the criteria needs to be well-developed and thought of, and there needs to be an empirical basis to our approach. So what are our methods? First of all, we're doing deep and wide consultations with experts with our newborn screening working group at EURORDIS, where we have amazing people with a lot of knowledge, and with experts from the Screen for Care Consortium. Like I've showed you, really talking about a very large and diverse group of people. Questionnaires will also be distributed via the EURORDIS Rare Barometer program. So we're talking about a program where we have about a pool of about 15,000 respondents worldwide that are members of the rare disease community. So we'll build a questionnaire in order to look at the different consideration that needs to be taken when we think of action. So again, the idea is to have a tool that is flexible and that different organizations would be able to use, also based on their position related to the different aspects. So examples of criteria that can be considered. We have already started working on a kind of framework paper, which kind of defines the different approaches that we can have and the different problematics that we need to address. So here are just examples. So should the diseases be added to the actionable list based on their severity of the disease, and what is the severity? Is the severity early death? Is it a mobility issue? Based on the prevalence, are we talking about ultra rare diseases or diseases that are less rare? What's the approach in terms of false negative and false positives? Whether the age of onset should be a criteria. So there is a big issue about screening for diseases that have an onset is definitely when it's adults, but when it's later on as well. Whether the damage that will be done if we do not screen is reversible or not, and whether it can affect reproductive choices. So here, for instance, question of the age of onset kind of are in relations to the question of reproductive choice. So if I take a late onset disease, one could say, well, this person can decide to know about this disease when he or she are older, but then it takes away from the reproductive choices of parents deciding not to have another child with the disease. So it is very complex, and we can do this game with all the criteria. Okay, back to Screen for Care in general. Screen for Care is about really the whole of the rare patient journey. And the idea is to turn the Odyssey into a journey. So we talked about the approach to at the level of birth, and then we have early onset of diseases, where we're really going to apply a more database analysis, and at the stage of the healthcare provider cycling. And here we're going to develop what is called a meta-symptom checker and a virtual clinic. Again, the idea is to provide solutions to shortening the path to diagnosis through the different stages of diagnosis. So now I'm going to talk a little bit more about the early onset and the part where we're talking about the data. Okay, the data solution number one that we're talking about, and here I'm going to talk less about the details because it's very complex and it's still something that is being developed. We're talking really about algorithms that will be used on electronic health record in order to identify and flag rare disease that could be seen from the electronic health record. The idea is to use databases that we had identified in the first stage in order to train the algorithm and then pilot it. What is important and very important for us as well at EURORDIS is that all of this will be done in co-creation with the patient, meaning patients will be involved in all the stages of the development. And it is very important for us that anything that is done is being done with the patient at the center. The second type of data solution are the virtual clinic and the meta-symptom checker. So the idea is to build upon what's being done. So we're talking about symptom checkers that already exist and see where the gaps exist, and also see whether we can federate the symptom checkers in order to turn it into a true point of meeting between the healthcare professional on one side and the patient on the other side in order to understand better their disease. It's especially important when we're talking about rare diseases because of the complexity and the need to base ourselves on data that is often pretty scarce. The virtual platform is really supposed to be a collaborative space for patients, families, and healthcare professionals, really one-stop shop where patient and professionals will move from a suspicion to diagnosis, to diagnosis and support post-diagnosis. This as well will be co-designed with patients, and EURORDIS is involved in all the stages of this task as well. Here we're talking about the virtual clinic. So the virtual clinic includes the meta symptom checker, but it is also a place where we can have communication room, real-time chatbot and texting. So we have one partner who is involved in, who is expert in this kind of virtual reality and virtual worlds, and they will create this interface. It's still in the making, so I can't give you details at this point. It's further along in the project, but here again, it will be co-designed with patients. We're going to pilot it, and we really hope that that will make it more applicable and also more sensitive to the needs of patients. So, just in terms of Screen for Care in general. So we are talking about a huge range of approaches or techniques that we are using in order to shorten the path to diagnosis. We have experts that are focusing on the data sides of it. So we're talking about utilizing existing data sources and registries, which we are mapping as we speak. We also have the newborn screening side, which is pretty expensive with an A. We're talking about really, as a pilot, challenging the definitions of the treatable diseases and the kind of, how would you say, the kind of conservative approach that diseases need to be, that we need to consider each disease at once, and that we should only look with what could be cured at birth. So the idea of promoting the actionable approach is really to broaden the scope, think of the family, also think of screening as an opportunity to do more research, and really think in a way that is broader and includes the patient, not only includes the patient, but puts the patient at the center. Screen for Care has only started, as I said, a few months ago, and we're still developing many aspects of it. You're more than welcome to get involved, to ask questions about what we're doing. We are talking about very complex processes, and we're really happy to be involved with the community and see how we can work together. As I said, we're very much in the community, and it is important for us to be part of what is being done throughout Europe and not invent the wheel altogether. So thank you for allowing me to present today. It was really a pleasure presenting Screen for Care for you. And you're very welcome to get in touch.
Thank you so much, Edith. And we've had some really interesting questions coming in. And I think the point you brought forward about, actionable and treatable is really important, because it's showing that even if there isn't a cure immediately, there's always something that can be done for these people and their families. And that's something that we really find to be important here. So thank you for that, and we'll see you at the end again for the Q&A session. Next up, I would love to introduce Pablo Botas to the main stage, who is the CEO of Foundation 29, and he's going to be talking us through a bit more of an AI approach, which is allowing patients to have access to and control over their own information and data, and he'll be talking about why and how data should be under the patient's control. So welcome, Pablo, and over to you.
Hi, thanks a lot. It's a real pleasure. I hope you can hear me well. If not, please just let me know somehow. So I'm going to talk about what our work here at Foundation 29, and the reason that we are doing what we're doing is due to our need to basically help patients living with a rare disease. And the goal of this presentation is to talk about why and how data should be under the patient's control. We believe that's very important, so I hope I can convey the message to you as well. So this is me. I'm Pablo Botas. I'm the CEO of Foundation 29. I also lead the scientific side of Foundation 29. My background actually is in physics, and I used to do a lot of research in terms of applying particle physics to cancer. And I'm working on Foundation 29 in the project that I will be describing shortly. So who are we? Who is Foundation 29? We like to define ourselves as rebels, and I know that's a little bit of a weird definition, but this comes from the fact that we really believe that patients should be the owners of their data, and they should hold it and be able to share it however they want in order to make better health decisions and in order to boost research. With this belief, we worked mainly on 3 different projects, plus a 4th one that I will be introducing today. And what I will try to do today is actually to tell you our story. And with this, my goal is to walk you through also our learnings, our key learnings, and how we fought against them a little bit also, and what we did to try to, and what we do to try to help rare disease patients. So mainly we have 3 projects, which is the X29, which is a rare disease diagnosis support system, Health Data 29, which is a site where patients and institutions can share safely data for research, anonymous data for research, and Health 29, which is now evolving to a platform that we call Raito, which is a data platform for patient groups. It's essentially a registry. So let's jump on the first part of the question of why patients should have their data. Well, you know, rare diseases, you know that there are many of them, 7,000, 8,000, it depends how you divide them. And that means that 1 in 17 people are affected by one of them. And it makes up for a global, huge problem in terms of healthcare, because you have to care for all these patients. And more often than not, you don't know really what's going on. So, you know that 75% of these diseases affect children. Actually, 80% are genetic, and many of them sadly don't make it to their 5th birth date. But what's really important here, and this goes to my point, is that 95% of them don't have an approved treatment, and it takes roughly 5 years to get to a diagnosis as an average, which is a very long time of going through one doctor to another, not really knowing what's going on. And patients without a treatment or without a diagnosis to begin with, because without an accurate diagnosis, there is not an accurate treatment, they are simply lost. And this is sad for many reasons, but it's especially sad due to the fact that there is a lot of information that can be harnessed, a ton of information. It's simply not being utilized. Patient data is underutilized, and it's underutilized for many reasons. But the most important reasons is data centralization. Data is held in what we call silos. Some silos are better than others, obviously. Not all animals are created equal, as this George Orwell book says. And essentially, these silos, these centralized places where data is stored and it's not easy to get out, act as roadblocks for scientific research and also for better healthcare management. So our goal is just, let's try to change this. Let's try to remove these roadblocks and empowering patients in order that data and knowledge can be generated in an easier way. And this is just something necessary to unlock the future. So let's go to the most important part of how can this be done, right? Well, first of all, you need to empower patients. You need to empower patients so that they can share their data in order to ask the following questions. How can patients leverage their data and their knowledge to improve their own health management? This goes towards a diagnosis, for instance. And also, how can patients boost drug development? Remember the 95% of rare diseases that don't have an approved treatment, right? So how can patients help in doing that? So I'm going to focus on the first piece, which is better healthcare management for a particular patient. And let me talk a little bit about what's called the Diagnostic Code. This is a graph that I essentially copied from EURORDIS, from Goldstein's work, which is a much better looking Diagnostic Odyssey, but I needed to adapt it to my purposes here. This is just a journey of a patient, right? From the beginning, when the first symptoms appear, to the first visit to a primary care physician, some online research with desperation, you know how this works, visits to specialists, then another one, maybe some wrong treatments, maybe some unnecessary surgery at some point. And more often than not, they reach the step of the genetic test, and there either you get to a diagnosis or you don't, right? So the goal, if we are trying to make this easier, would be something like this. From first symptoms, you reach quickly to specialists, and then you go to diagnosis. And the question is, can patients have a more active role in this process? And one aspect that patients do know about is themselves. And they can describe themselves very accurately, right? Maybe some terms are not clinically perfect, but they do know what's going on with them and what symptoms they have, right? And coming up with very accurate descriptions of the patient's symptoms is very important. It's also hard, but patients really can help there. That's what we've seen. This is just a phenotypic description of Dravet syndrome, which is a rare form of epilepsy, and it's very severe. So this is something very important, to have a proper, detailed and accurate description, phenotypic description, so in terms of symptoms of a patient. But even when you have that, it gets complicated because most physicians might not know this disease, especially primary care physicians. And rare diseases in general are affected by this. They have complex phenotypes with many symptoms that are shared between diseases that have gradual evolutions, and then the differential diagnosis becomes very, very tricky, and it depends on experience, but not a lot of physicians have this experience, so there is a gap of experience, information, and also a big gap in access to optimal genetic test and analysis of the results. So the goal that we took was like, well, can we solve these gaps? Can we facilitate phenotypic descriptions of patients? Can we then, based on these descriptions, predict what diseases are candidates for these particular patients? And can we do that all in an easy solution that physicians without a lot of training and patients with no training essentially can use? So we looked up out there, and there is good news in the rare disease community, and is that given its low prevalence and the severity of these diseases, there is a very interesting movement. You just have to see at this session, for instance, how people are really innovating out of the need of creating solutions and new quality of life for their community. So there are some resources that we can touch on, and we know that there is disease information. There are some resources, mainly Orphanet and Omnium, that have done an amazing job collecting and specifying the phenotypic description of many diseases. And by this, I mean lists of symptoms, sometimes even frequency, how often a given symptom appears in the community and how often another one does. We know what a symptom means. That means we know that a focal seizure is a type of seizure, which is a type of, etc. And we know that because the guys from the Human Phenotype Ontology created this amazing resource now describing, I think it's 17,000 different symptoms, which is amazing. And we know also, thanks to some data sharing projects, mainly ClinVar and now ClinGen, about the genetic traits of many diseases. So based on these three pillars, we created what is called DX29. DX29, it's a completely free website. It's open source. We just created it to demonstrate how the problems that we saw in the Diagnostic Odyssey can be solved through technology and through collaboration between physicians, patients, and other stakeholders. DX29 is accessible here, and I invite you just to check it out. It's for physicians and for patients, and you can just try it out with a registration or register as any of these roles and start using it. It's very simple. It basically allows you to find symptoms in your own medical reports. So if you have a document sitting on your desk, you can take a picture with the cell phone, upload it to the X29, and it will tell you what symptoms appear in there. And that works in 50+ languages, which is pretty cool. And sometimes it makes mistakes, obviously. So you have the power to say, yeah, you messed up here. This is not correct. Or you have the power of saying, you missed this one. Please add this symptom because I know I have it and it's not in the list of symptoms that the tool has identified. Based on this information, and you can also add genetic information if you have that, although I doubt it because not many people have this genetic information on their laptop or cell phone. Based on this information, it can predict what rare disease you might have, or the information that is provided points to. And you can create reports based on this. They are just informative. They are for you to take to your physician, for a physician just to check out. It's important to know that obviously it can make mistakes, and it said on the report that, hey, just take this. This is just information. Here you have the evidence that it's based on, and here you have a preliminary list of diseases as suggestions for your physician to check out. So this is how it works. I'm going to show you briefly a bit of the steps. If you upload a document that is not in English, you will have the original language here, and then all the entities and symptoms that are identified, you will have them there. And we use 2 different algorithms for that, one from the Sick Kids Hospital in Toronto, with whom we have a collaboration, and a proprietary algorithm from Microsoft that is also working pretty well. As a result from this, you would have a curated list of symptoms, curated by the user, I mean, list of symptoms. Based on that, we can predict what diseases you might have, because there are some information that was initially created by Orphanet and OMIM and all this human phenotype ontology that I mentioned, right? And based on that, we can predict what disease the patient might have. And if you have also genetic information, it can be read and compared with the symptoms to pinpoint what a specific mutation might be the cause of the patient disease. As a result, you will get a list of diseases and with a brief description and a list of symptoms that will inform you of the symptoms that the patient has and the symptoms that the disease has. So you can make a decision, or the physician can make a decision on what disease is more plausible, is the most plausible explanation for the patient. So this, as I mentioned, is powered by Orphanet, by the guys at the Monarch initiative that created Mondo, the human phenotype ontology, and Exomizer, and algorithms from Microsoft currently. So key results of this initiative. Just first of all, some statistics to give you the background and the ballpark figure of how accurate this is. Based on roughly 800 patients, we can predict in the top 10 of the suggestions the correct disease in approximately 50–60% of the cases. Okay, this might sound low, but it's actually pretty good, and it's on par with the most modern algorithms out there. If you add genetic information, this number goes up to 78%, but in the top 5 of the suggestions, not in the 10 first, but in the 5 first. So it's much more accurate. If you have genetic information from the parents, the 78 goes to 98%. It's much better. And the key thing here is that actually the X29 is effectively helping closing these gaps between the expertise of the physician, maybe the lack of the phenotyping capabilities. And we've learned that patients doing this work of uploading medical reports and selecting what symptoms are correct or what symptoms are not, they reach very precise phenotypes. So they can be trusted by physicians, at least in the first iteration. Maybe they can be refined sometimes. And just to give you a number, we have approximately 130 daily users. It varies, obviously, but that's approximately what we have. And now some important learnings, because we did this and It's working pretty well. It has helped already 5 different departments and hospitals to improve the diagnostic odyssey for patients. But the reality here is that in general, specialists are too busy to be engaged with this type of systems. And general practitioners are more collaborative. So the last resource that you actually have in order to do this, because healthcare information systems currently are not as agile to adopt this sort of technology. So if you want to push for this technology right now, you need to go through patients. Otherwise, you need to move whole healthcare systems to adopt this type of things. So this is the bottom line. For any of these to work, you need patient data. Patients need their data. So this brings me back to this previous slide, and I'm going to talk briefly about how patients can boost drug development. And the reality is that drug development for rare diseases has not evolved as good as we would like to, because if you see this plot, it looks like many drugs are being created. This is orphan designation. The graph ends 5 years ago, but it hasn't changed that much. And you see that it's growing, but actually authorization is not growing that much, Parima. And this is a problem. And also a lot of these diseases, of these medicines then end up treating some diseases that already had a dedicated previous drug. So there is a big need here for a data-driven boost, and patients have the key. The underlying truth here is that we are not learning quickly enough. And the reason is what I mentioned before. It's this centralization. It reminds me about just a gravity field around the Earth where satellites revolve around. And hospitals, registries, and trials and other entities obviously have the same effect on data. Clinical data somehow always ends up around these entities, and also patient-reported data. And this is great, but the problem is that they are not able to share it with the community for many reasons, mainly because the data is not theirs and they act as custodians. Sometimes the business model revolves also around that, which doesn't help. So the key here again is that patients own this key. And patients also have a gravity field, and anytime they go to a specialist, there is some data generated, or to a physician, there is some more data generated. And before there was a technological barrier to actually try to gather all this data and make it useful and share it effectively and so on, but there is not anymore. There is not anymore. There are different standards that facilitate this. And I'm just gonna go really quickly through our next project. And the idea here is not to showcase the project, but to explain how it works, how we think it can work. It's an example of patient registry 2.0, if you will. And the 2.0 comes because it's a decentralized patient registry. So we call it RAITO. It looks like this. It's very simple. It's essentially patients get the power to manage their own disease and to boost drug development. And they get this power because they get to own their own data. And they can track different parameters in their life. So for instance, seizures, quality of life, the medication they are taking, and so on, and visualize how all this evolves. And they receive questionnaires just to provide patient-reported outcomes. And these eventually will facilitate drug development because it facilitates the demonstration of significant benefit for EMA and so on. And this is the important key. The data is not stored in RAITO. The data is stored in the patient's device. It follows FHIR, which is an international standards. It follows international standards also for sharing. The standard is called SOLID, it's just the detail here. And that's the important key. In RAITO, we do not store anything more than just the registration data, like name and email of the patient, nothing else. Everything else is controlled, owned, and shared by the patient wherever they want. They provide access to some anonymous data to their patient organization, and then the patient organization can aggregate this data, and they may provide access to a different research project that they find interesting. So just to wrap up, I know I went pretty quickly here. I hope I was able to convey my message. The key aspect here is that if you want really to improve rare disease diagnosis, if you want to improve the development process of rare disease treatments, you need to accept that data centralization is a roadblock. And actually we came to the conclusion that it's not a good idea to invest in some project that will end up with data centralized. Data should be decentralized in order to flow effectively. There are currently standards that allow patients to take control of this and to share it in a secure and ethical way. So patients need to be in charge of that, and they can actually change this. So nothing else from my side. Again, it's been a pleasure. It's been a pleasure also listening to the previous speakers, and any questions, I think they will be answered at the very end, correct?
Yeah, correct, Pablo. Thank you so much. It's brilliant to see how you're really innovating to bring people the answers that they've been looking for years, and that hopefully we'll have some interesting questions for you right at the end. But now, last but not least, I would love to welcome David Issam onto the stage, and he's going to be talking about designing empowering digital therapeutics and a patient-led approach to that. So over to you, David.
Hello, thank you for the invitation and good afternoon, everyone. So I'm David Issam. My background is in digital health and global health, and I'm the lead of the research at the ESCF, which is the European Sickle Cell Federation. It's an umbrella organization which mission is to engage all patients in Europe to provide better care, better access to services and quality of life. But today I want to discuss more on the challenges and the opportunities of digital technologies to support such missions and see how we could accelerate healthcare transformation. So first of all, I will go towards a small introduction on the general context, and then a small description of the specific case of sickle cell disease, and then dig a bit more into the technological aspects. So, as you know, we are living in a world full of health inequalities, where the resources such as education or research funding, access to treatments, political rights are not distributed equally. And this regarding the geographical and socioeconomic context, it will differ. But there is no fatalism to that, because inequalities can be reduced using the right mix of action. But acting is very important, because you know that inequalities are increasing, and as the COVID crisis has shown, people with rare diseases, or what I prefer to call forgotten diseases, are particularly affected. And I will take the example of sickle cell disease, which is one of the most forgotten diseases. It is a genetic blood disorder, which I know myself very well since I also have it. And it's a systemic disease because it affects the body in various ways, in various places, and it creates different complications which are acute and chronic. As well, it's a good example as magnifier of health inequalities, because despite an increasing prevalence and quite numerous people in some places in the world and less in other places, the burden is increasing. People have a shortened life expectancy, about half less in the high-income countries, and actually most people in lower-income countries die before their 5th birthday. And by consequence, you have big loss in quality of life, productivity, social participation. People struggle with mental health issues, for instance, and all that has a big societal cost, financial cost, but also unemployment, misinformation, and various issues. But basically, people face poor access to quality healthcare. We know that there are really few specialists for the disease, also some stigma, little research, poor access to treatments, and little treatments. And in consequence, people have to rely on themselves to live an as normal life as possible, which is called self-care, basically, and consists in different things such as risk factor prevention, better monitoring our symptoms, understanding how to conduct a better lifestyle, all the practices, all the attitudes that can be relevant. So they may struggle to keep positive attitude towards life when they would suffer so much, and struggle to attend medical appointments and being followed up, since they may also mistrust healthcare providers. And so we know that a little number of people are able to fully self-care, but hopefully there are some strategies to help empower patients, and in particular, digital technologies can have a huge role to play to reduce the burden on patients. Empowerment basically means the efforts an individual can make to control and change their destiny by learning better skills, learning how to better interact with people, with the environment, and then in the end have more power over society, so society can provide better societal and social resources. And digital technologies are, as I said previously, of big importance for that, because now we are at a stage where we can have sensors on ourselves. We can have sensors outside on your environment, for instance, for the weather, and gather all these different data sets to predict health outcomes, to guide better decisions. And now into this field of digital health is the field of digital therapeutics, which are, as you may understand, like drugs, which could be prescribed as a therapy, but digital. So these are like software which are evaluated and validated, so they can support people with disorders. But there is no one-fits-all systems, even for sickle cell disease. But there are many inspiring ones, like this one, Gaia Health helps for musculoskeletal pain. You have systems which can support substance use disorder, software to support cancer care, to alleviate respiratory diseases, diabetes also. Many apps for diabetes, also for mental health support, also including, for instance, CBT or mindfulness interventions. And also importantly, in the case of sickle cell disease, systems which can support PTSD, which are also evaluated, but this is still a niche for now, and there are many challenges related to democratizing digital health. Like as it has been explained previously, interoperability is extremely important, because we need to be able to share data, to exchange information. If not, we will just have various silos and systems that cannot be compatible with any other one, nor communicate. There's also big challenges towards reaching strong evidence on the usefulness, on the effectiveness of the systems, and importantly on the adoption, because it's okay and it's good if you have an app, but if you don't use it after 1 week, then it may not be so useful. And we know that there is an average of about 1 week of retention in app use, for different reasons, such as overlooked patient needs. For instance, if you imagine that devices could be created mostly for people with whiter skins, it may not be compatible with people with darker skins, and these things could prevent people to use the systems, and other costs for instance, cost of acquisition, cost of sustaining the system using it, and for instance, of course, environmental sustainability is also important. So to try to go further, to go towards these challenges, we propose to switch paradigm to leave the patient-centered approach, where in general the designer or solution maker will see a problem but also a solution, and move to patient-driven, patient-led approaches, where the patients themselves, they leave the problem, they see what should be the focus and how the problem should be solved using all the realities to have an as precise as possible solution to their unmet needs. So such approach are very important in the rare disease world because it's often less well funded. So basically it is about giving patients the steering wheel so they can create themselves the solutions that will help themselves. So how we do that? Well, first we discuss with people, we understand their needs, we let them prioritize what they want to have solved. For instance, having helped to early detect health deterioration, having systems which are simple to use, for instance, or very discreet in their life, in the sense that that will not remind them of their sickness and add additional burden. So we then can think about how to meet people where they are, adapt to their context, and be as discreet as possible. We know, for instance, that most people use messaging apps. So it could be an idea to have a system that work into messaging apps. And then it is about co-creating with the people what they will want to use, because it would solve the issue. And technically, to do that, we can leverage state-of-the-art technologies such as crowd sensing, which is the use of various kind of mobile technologies or edge technologies, how we can also say. Use also design approaches that will persuade people to use the system, and then have a proper use of data interoperability in the sense of standards that will allow then further use of the data generated by the patients, and for that enable interpretation of data, exchange of data. And also, as it has been mentioned also previously, it's important to keep the ownership of the data to the patients. So the regulatory aspects are also crucial in that aspect. So this is an example of this approach and what we have done using such vision. This is a companion which people can use to share data about their daily life and create a registry which can then be further used to understand better how people live. So the idea is to enable people to share where they are, what they do, what are their physiological data, for instance. Based on that, we can also understand what are the struggles, where people are, how people are empowered, where they should have more help. And by leveraging patient experiences, we can then target what works best for some people, in the sense that we can propose them advices on what drugs, for instance, works well for some specific subsets of patients. And of course, provide them with therapeutic education in the sense of helping them to better understand why something works well, and follow them step by step, and encouraging them using also peer support or discussion groups, so they can then, for instance, have mentoring and discuss on how to implement better habits in their daily life. So this system has been evaluated on the usefulness and the usability, which was quite well received by the patients. Now it's on the effectiveness evaluation side, still ongoing work, but we seem to see that such approaches could accelerate a shift in mentality in the healthcare world, and therefore bring solutions to patients faster, and while they are also more fitted to patient important needs. So some advices we have from that is to start with people who struggle the most, because their realities will be very complex, and they will be able to see where there are, for instance, bottlenecks and things that people who are better off will not feel or live, for instance. We also see that it's important to think about all the ethical issues of such do-it-yourself systems, because it will bring new challenges. And it's so extremely important to make sure that the data that are created can be further used. And I won't take more time for now, but we can discuss later on, and I will be happy to hear your questions. Thank you for your attention.
Thank you so much, David. It's wonderful to see the kind of co-creation approach that you've taken there. And I would love to bring back all of the speakers to the main stage now to answer some of the questions that we've had coming in through the chat on the platform. Okay, great. So we've just had a question come in now, and it's the latest question, but I'm going to start off with it because I think it's quite a nice one. So somebody has asked, how accessible are newborn screening approaches and technologies, and are there projects in place to bring more of these approaches to African patients, for example? This is something that all of you might be able to kind of give an answer to. So whoever wants to unmute and go ahead first, I won't pick on you.
I'll start, for instance, since I'm not muted. So you're asking about newborn screening, and I cannot really comment on that. What I can comment is, for instance, DX29, which was one of the projects that I highlighted during my talk. The Ghana Rare Disease Initiative is now testing it, basically because since it's open source, they can just take it and put it wherever they want, and that makes it very easy for them to close these gaps that I was mentioning. I have to say that there might be unexpected biases, as David mentioned, because rare diseases may present differently on African population from that area than from human population from other areas, and that we don't know. That's something that needs to be tested yet.
I think I could comment from purely sickle cell screening perspective. So, I mean, the methods are accessible and they can be set up, to reach African patients. And I think, what I try to talk about simplifying workflow and analysis. So, of course, we are also driving purely for the sickle cell screening for solutions that are sort of automated data handling and interpretation. But in terms of, is it accessible? So it is possible to bring the screening technology, and where we have programs running.
Great, thank you, Pablo and Mikael. And then we had a question right at the beginning, which I think was directed at Marie-Christine. And somebody is interested to know if SMA Europe has a registry. And also, I would be interested to know, if you do have a registry, how do you connect all of the data you're gathering across your newborn screening programs? How do you bring all of that together and actually use it for the most efficient purpose?
At this time, there are different type of registry. There will be, we are focusing about the number of babies screened in Europe and the number of baby positive at the screening. So it's a sort of epidemiology regarding SMA to see if it is similar what we know in the general population. And but after that, when the baby is diagnosed, it goes to the National Registry of SMA, because there are existing registry for SMA, and it's part of the National Registry of SMA. And there's a big database owned by TreatMD or driven by TreatMD, who is available for SMA. So we have just focusing about a sort of epidemiology to see if the diagnosed population via newborn screening is more or less similar than the symptomatic population.
Thank you, Marie-Christine. And then we had another question early on as well about, and the person has asked, based on your range of technologies when carrying out newborn screening, would you only be screening for selected known disorders, or could you potentially find previously unknown conditions? And I think this was during Mikael's talk, yeah.
Yeah, I think so my answer is that typically in newborn screening programs, you really have a conservative approach on what you screen for, and even saying that you are not willing to have secondary findings. So I would say, like, this is a typical concept of newborn screening. You're looking for a certain disorder, and you can sometimes even look at these secondary findings as false positives. However, we have screening programs that look at it in another perspective. When they screen, they have screen positives and they have secondary findings, and they look at this as that cases that will be researched further. But I think purely to make a case for implementing new disorders, it is quite crisp and clear what you're trying to look for.
Great.
Others might comment if you have a other opinion.
I don't know if anyone. If not, I've got a question for you, Edith, regarding the Screen for Care programme. Someone asked, will that only be accessible for the nationals within the countries, or open to external EU patients through the S2 scheme?
Okay, so it's important to understand that Screen for Care is a research project. So at the moment, what's going to happen, I guess the question is around the newborn screening. So the screening will be done in those centers that were chosen to participate in the study. And so I guess this is where things are going to be done. Of course, whatever we learn and the output and the deliverable will be available throughout. But yeah, but it's a research project. I don't think it distinguishes between national and others. It's just that it will be in those selected centers.
Great, thank you, Edith. And then there's a question here which is quite interesting, and I think will probably be able to be answered by a few of you. And somebody's asked, is there a relationship between developing newborn screening programs and the normalization of the storage of umbilical cord blood? If not, should there be? I'm not sure who is best placed to answer that one.
I guess it's a tough one, because the question is whether it's a sociological or anthropological question about people's habits and how open they are to having information and having their samples being out there and to use for the future. And if it's a question, I guess, clinical questions, then the uses for newborn screening samples is very different from the uses for umbilical cord. So it sounds like it's in the same area, but it really, I don't see how we can directly link them, other than the fact that the two belong to an area of medicine that is precision medicine. And
yeah, I would also agree on what they said here, and in a way, I mean, the power of newborn screening and some of the programs is, in terms of like biobanking, is the DBS format, and the DBS sample can be also, you know, interesting and holds holds very well, but I don't see these two different sample matrices actually like coming together.
Great, thank you. And then kind of something that I was interested to know, and Rick was interested to know, particularly for Mikael, is so obviously after diagnosis, it's really important that patients get a diagnosis, but what about the way that is delivered, and how does the whole of this come together so that the patient feels supported after they've gone through that process of newborn screening and have got the answers? How does it all kind of come into one holistic model, or is that something that's still being worked on?
I think that is exactly the point where, in a way, we're again talking about from country to country, to region to region, what Mara-Kristin also was explaining in her presentation, and we sort of touched the screening and sometimes the diagnosis. But for me, the question is more like if you can screen for it from technology perspective, probably it's easier to set up the screening assay than to set up the genetic counseling, the treatment, and the follow-up path. And for that part, I understand that even my part of visibility is quite narrow, and it just highlights that we need to start from connecting with all stakeholders and to get also the downstream path. And I'm looking at the part in the middle. I don't know if I answered, but
Thanks.
that's how I see it.
Yeah, no, I understand. There's still there's never finished with just one aspect, is it? There's still so many things to cover. Marie-Christine, I see that you've unmuted.
Yes, just the screening test is a small part of a newborn screening. And for example, we are on the way to have an ENMC meeting to more or less define the best practice as soon as a baby is screened positive. At this time is different from one country to another one, and we want to select the best country, how to announce to the family what is the best treatment we can offer, what type of support, what type of document you have to give to the family to better understand. But yes, it's another part. At this time, more or less in different country, they are sent to a center who are used to treat the SMA patients. But there's a step between the diagnosis and the start of a treatment that need to be more investigated, I think, and more taken in account.
And maybe to add that, I mean, there are guidelines like CLSA guideline, and there is a SMA newborn screening guideline being worked at in the CLSA, which has a global perspective, and this will sort of have a picture of the whole approach from sampling and going into all the way into communication and treatment.
Yeah, and just to add to that, if we've mentioned Dr. Gulcin Gumas a couple of times in this talk, so Gulcin has developed, along with the newborn screening working group in EURORDIS, this 11 principle for newborn screening. So one of the ideas of these principles is really to have a full support for diseases that are screened, and that this should be something that should be taken into account when selecting newborn screening. So I think it links very well, I'm sure, to the work that Marie-Christine is doing at SMA. So it's great that things are kind of coming together.
Great, thank you so much to all of you. And then a question specifically for Pablo. Somebody asked, is there a way for researchers to send a message to the patient, albeit anonymized, if they find anything worth notifying?
I guess they mean in DX29 in the platform I presented. Not through DX29, but there's also pretty much no situation where a researcher does not know who the patient is. Either they created the case or the patient has shared the case with them, because that's something that can be done within DX29. And that means that they have the contact information of the patient for that. So it's a no within the platform, but they have other means. Ultimately, if what you want to try to create, or what you want to create, is a demonstrator of how something could work, they're usually no, not end-to-end platforms that would allow this type of communication and so on.
Great, thank you, Pablo. And I kind of had a question as well, which was, do you find that people naturally feel safer holding their own data and kind of secure within their accountability towards that? Or do you need to convince people that, like, having this is a positive thing. I know that if I had access to all of my data, I'd be worried that I would lose it, I'd forget the login, and then I can't blame anyone else if that happens. I don't know whether you found this to be a thing, whether actually people, if they can give it to someone else, at least they're not holding all of that responsibility, or has that not been something that you've experienced during kind of setting this up?
Well, it depends a lot on the person. So there are a wide range of feelings there. One of the key things to say is that having patients with your data doesn't mean that it's the only copy. So for instance, within the NHS strategy for data managing, they envision going in this direction, but for some time having essentially 2 copies and having the patient empowered with their own data, while also keeping a record at NHS. And this allows the patient to effectively share it with whoever they want. So that would mitigate that. But it's a very good question. And it depends on the profile of the patient, on how used they are to this type of things. It doesn't mean that you have all the responsibility at all, but there are mixed feelings.
Great, thanks, Pablo. And then somebody else wondered where individuals can actually get their genetic data if they wanted to feed that in.
That's a good question. So for instance, in Spain, if you went through a genetic test, you have the right to request this data, and you can do it at the hospital, at the patient office. And then the hospital usually enters in a situation of chaos, and they don't know how to give you the data. So they end up contacting the lab, and they end up just figuring out how to move the data from the lab to the hospital to the patient. And it's a bit of a mess because we as citizens have that right, but it's not a right that we actually use. In other countries, it's similar. And many patients, given the difficulty of accessing to genetic testing, they just go to a lab and do it themselves, in which case they already have the data after the test is finished.
Yeah, great. Thank you, Pablo.
Yeah.
Edith, I kind of had a question for you, and I hope that I've got this right in the way that I formulated this question. But on the screen, you obviously had kind of examples of criteria that you're using to distinguish between treatable and actionable. And I wondered how you prioritize between all of those, because you had like severity, some other ones, and also how you're deciding on those things such as severity. Is that coming from like patient voice, quality of life questionnaires, and who's feeding those into you to actually make those decisions on which criteria you're going off?
Yeah, so I guess you're touching exactly on the main issue and why we are on what we're going to work for the next year and a half. Exactly on thinking of what are those criteria and developing a better understanding of the different criteria. So I just put those on to show kind of things that we're considering, but these are in no way criteria that we've settled. And what we're going to do is really have consultations, so with the newborn screening working group experts, with the consortium experts, in order to build from that. So the idea is to have a deep ethical work on those criteria, define them better, get in touch with other groups that are working on similar things, such as Genomic England, and then get the questionnaire in order to get empirical data from the rare disease community as well. And at some point, I believe what we'll be defining is not really a prioritization of the criteria, but more of a conceptual scheme, a framework, where different organizations will be able to use them as a starting point to consider what diseases should be included or not included. And it also help at the level of the issues of secondary findings and so forth. So what we're aiming at is really have some kind of tool that is flexible and sustainable, and definitely not something that is prescriptive and where we've made decision on what is more important. That's a value-based decision, and of course depends on the context.
Thank you, Edith. And David, I just wanted to ask you a question as well. I was wondering if you know of any other groups that are using this kind of technology that might be doing a similar thing, or whether you think it's an approach that everyone can use in the rare space, every kind of patient group, or it's very much community driven on whether it's going to work for you or not.
Okay, good question. Basically, the thought behind has been to be as generalizable as possible, because in the context of empowerment, we have defined two main concepts, which will be functioning, like social functioning, physical functioning, etc. All the things that will make you able to live your life, and then the concepts of readiness, and that will be all the information we can collect to tell a system or to tell other people that you are ready to have a good day today, for instance, or to act, to go to school, etc. So basically these concepts are extremely generalizable, and on which we have based the system based on this concept is usable for any disorder, basically. And today, my knowledge is more advanced in diabetes world, but other subset of diseases are also catching up. I don't know if I answered well.
Thank you so much, David. So I think it's about time that we start wrapping up this session, but I'm really grateful to all of the speakers today. I've learned a lot, and it's not something that I'd previously actually been exposed to that much. So thank you for such clear and informative presentations, and for showcasing some really exciting things that are happening in the space as well. So I hope to see some more of you attendees in today's afternoon breakout sessions as well. There's still space for the funding research in low and low middle income countries session, and it'd be great to see some of you guys on that session with Cures Within Reach. And then this afternoon at 5 p.m. GMT until 7, we've got a look at the rare diseases across the Americas, which is a really exciting, another kind of location-based look at what's happening in rare diseases across that region. So it'd be great to see you in that session as well to wrap up day 2 of the International Rare Disease Showcase. Once again, a huge thank you to all of our speakers, and I'll see some more of you later on this afternoon.